Drew Harvell is a professor emerita of ecology and evolutionary biology at Cornell University, affiliate faculty at the University of Washington, and a former science envoy to the U.S. State Department. She is the author of Ocean Outbreak and A Sea of Glass. She has written for the New York Times, the Seattle Times, The Hill, and CNN, and has authored over 180 academic articles in Science, Nature, Science Advances, and more. Dr. Harvell was featured in the award-winning film Fragile Legacy and is currently a science advisor for Fabien Cousteau's underwater space station, Proteus. Without further ado, please join me in welcoming Drew Harvell. Thank you.
We don't need that, right? Thank you, James. Move that aside. Welcome, everybody. It's really a pleasure to be here. This is probably about the best place for me to be able to give a talk like this because, well, Seattleites love the ocean and are very environmentally aware, and we just have fabulous biodiversity in our ocean here, and so that'll be my topic for tonight, how are sailish sea stars faring in our ocean's menagerie. And the other point I like to make from this slide is just the fabulous biodiversity we have, particularly in a group like the sea stars. It's a little hard for me to actually see you because of the lights, but I don't know if any of you know how many species we might actually have in our waters here, but it's roughly estimated at about 80 species of sea stars. Right? That's a lot. Now, that's not just what you'd find in tide pools and the near shore shallows, but of course that extends into the deep waters. But a pretty fabulous biodiversity, and so that's really what I'm here tonight to talk about, is to celebrate our oceanic biodiversity. And what I'll do tonight is I'm going to sort of set the stage for talking about the biodiversity of our ocean critters from the book that I'm just releasing called The Ocean's Menagerie, and then delve into some of our recent research findings on sea star wasting. We've been working on the epidemic, the impacts of the epidemic, the causative agent of the epidemic, and the recovery projects for the epidemic for well over a decade now. And so it really means a lot to be able to share some of that information with you tonight. Ocean's Menagerie, my book that's just out in April, focuses on essentially these 8 groups of invertebrates in the ocean. And tonight, because I'm going to be talking about some of our research, I won't go into all of that. But if you have the book, then you'll have a chance to read more. So I like to sort of just start by thinking a little bit about our history on this planet, and I do have a trivia question for you. I'm wondering if anybody knows how old the invertebrates are, in the sense of when did these groups originate? When did they evolve on our planet? And these are some of your options for an answer. And I thought what I would do is I'll go through each of those, and then you can raise your hands if you think you've got the right timing. So does anybody think that our invertebrates, and by invertebrates it includes the sea stars, includes the corals, includes the sponges, includes a lot of our seashells. Roughly when were those groups evolving? Do you think it's 34 million years? Anybody got an estimate of that? I'm not seeing any answers here. Now you have to be sure, everybody has to answer something, okay? So, how about 200 million years? Boy, no takers on this. Oh, there we go. Thank you very much. We've got one. 425. There's a lot of people out here that are thinking that makes a lot of sense to them. And what about 600 million? And those of you that said 600 million, that would be the best answer. Although for some of these groups, for example, the actual photosynthesizing corals, that may not have happened till 425 million years ago. So you guys weren't wrong on that. 200 million, that's dinosaurs, right? So what, 250 to 200 million years ago. So these are such ancient animals, and yet they are still in our oceans today. And every time I think about that, it makes my head spin a little bit, especially given how much our climate and our oceans are changing, and the phenomenal capacity of these critters. Now, I'm not as old as the invertebrates, but I have been at it a long time, and yes, I do still dive, and so this is kind of some of my early, how I became obsessed with the kind of work I've been privileged to do my whole career. As a graduate student at the University of Washington at Friday Harbor Labs. Have any of you been to the San Juan Islands and Friday Harbor? Yeah, okay, thanks. A lot. I was a diver then, and I was invited along as part of a research team to be part of a saturation diving project. And do we have some divers in the group too? Yeah, okay. So the divers in the group know that normally if you go to 60 feet, you might spend an hour at the most there. And if you went to 120 feet, you would not have more than 10 minutes there. But because this was a saturation diving laboratory, we lived on the bottom for an entire week, and so we could spend an hour at 120 feet to be able to do some really big surveys at those depths and down to 150 feet. So that was a pretty amazing opportunity for a grad student to have. And then I went back and did 3 more and eventually ran a couple of my own missions. And so I do kind of still feel like I have gills when I'm on a dive underwater, and it certainly changed my life. And that person in the pink there in the bottom, that was me way back when running my first team in the saturation diving habitat. And so it's been a fabulous career of studying essentially the ocean's menagerie. So this is the organization of this book, and the shorthand, I kind of call it the superpowers of the spineless, because my real goal in writing this book was just talking about the fabulous biology of these invertebrates that are so ancient and do things unlike anything on land. I mean, for example, we don't typically think of animals as photosynthesizing, and yet corals, through their symbiosis with algae, are photosynthetic, and then that's why it's called the coral's castle, and build these enormous castles. Tonight I'll say a little bit about the sea slug's sting. Again, that's an incredible feat biologically, really, to be able to steal an organelle from not just another species, but from a group of anemones that are way across the tree of life from them. And then finally, the sea star's sticky skin. So, and then these are just some of the other topics I won't have time to talk about tonight. So normally, one of the foci in this book is to talk about bio-inspired design from invertebrates. So it's a little bit of a what's in it for us kind of a story, in addition to, for me, there doesn't need to be anything else in it except the wonder of these invertebrates, and for the divers and some of you in the audience, that's probably the same. But there's a lot in it for us. And the examples I'll give tonight are cancer drugs from sponges, transplantation biology from nudibranchs, and then the sea star's sticky skin, when then I'll talk more about the sea star wasting in that project we're working on. Normally I talk about corals, castles, and jelly bioluminescence, but there won't be time. This is probably one of the best examples of just incredible riches from the oceans that have been so beneficial to humans. And early in my career, I worked with natural products chemists whose job was to find biologically active chemicals from sponges in this case, because that ended up having the highest hit rate in nature at the time. And the reason they wanted me along, because I'm an ecologist, is that I was really good at finding really rare and strange colored and ecologically weird sponges that had a lot of chemistry in them. And also because I was fascinated in what do they actually do with these chemicals that we're so interested in. This is just an example of one. This is a swinoolide, and you can see at each one of those little junctions there would be a carbon, so this is probably a 20-30 carbon compound, so a very big, complex biologically active chemical. This one can stop the growth of fungi. Cytotoxic means it stops cells from dividing and disrupts cell divisions, which of course, if you're interested in a cancer drug, that's what you want to be doing, is stopping in a targeted way the division of cancer cells. And the reason I highlight Falconer's study here, which was in the I think it was published in the mid-90s or early 90s, is he was the first to realize that these chemicals were not actually made by the sponges. Initially, we thought the sponges, because they were in the sponges. It turns out that many of these biologically active compounds are made by bacteria that live inside the sponges. So there's this whole other level of biological marvel in the dance between the host sponge, the particular species of bacteria that lives there, and then what are the conditions that sort of optimize the production of these chemicals, and of course then what are they doing with them in nature. And so I don't have time, although I talk about it in a lot of detail in the book, a lot more examples. I love this one, though. I don't know if any of you recognize that sponge shown there. That's Helicondria panacea. It's common in our intertidal. It houses a Streptomyces bacterial strain that makes a chemical called miamycin. And this synthetic derivative is currently used in the treatment of 5 different carcinomas. And in fact, it's pretty amazing to me, a few times when I've given talks, people have actually put their hands up and said, my relative or I, or we are on that drug. And it's actually pretty whiz-bang in that in treating bladder cancer, it's actually produced as a gel that sort of remains active for a very long time. A related species, Helicondria occidii, houses bacteria making helicondrin B. This is an anti-tumor in clinical trials approved by the FDA to treat breast cancer. So these are just a tiny, tiny example of some of the riches in chemistry that we have from sponges. And so in my kind of way of talking about this, I call this the superpower of the sponge group, is this producing incredibly active chemicals. Now, does anybody here like nudibranchs? Are there? Yay! All right, yeah, maybe it is my favorite group too. I mean, partly it's because I had my early beginnings here at Friday Harbor Laboratory, and we have I sort of went on and on about the biodiversity of sea stars, but the biodiversity of nudibranchs here is also really spectacular. So these are just a few of our local nudibranchs. I took these pictures at Friday Harbor, where I'm an active researcher and an educator, and these were just in our tanks for the students to study and work on. And I'm going to delve a little bit more into the stories about nudibranchs. So probably this is familiar to some of you. This is one of my favorites, and this is a video I took a few years ago watching this nudibranch creeping up on the sea anemone. And the thing that was interesting to me was how slowly it moved. It really crept up on it. And I'm not going to show the actual killing blow where after the anemone retracts, this thing actually pounces on it and rips it apart. But I often wonder about that slow kind of approach, and what you see the anemone has done is it's pulled in its tentacles, which is, of course, where the stinging cells are, and there's stinging cells shown in the top of that picture. So those are the harpoons that are characteristic for all of the cnidarians, whether it's jellyfish or sea anemones or corals or hydroids. And this is the sort of the superpower of the nudibranchs, is the ability to actually feed on these prey and then uptake those harpoons. And then this may look like the Easter Bunny. It's pretty and fluffy, but this thing is loaded with those stinging cells in those back projections on its back. And so again, in the book, I talk about sort of the 100 years of trying to figure out how this actually works. We know a lot more now. We know that, oops, I need to get past this somehow. We know that that the nudibranchs tend to select out the immature nematocysts, that's what those stinging harpoon things are called, pass them through their gut, and then there's actually projections of their gut into those back projections. And then the real magic, the real kind of superpower part of this is that the nematocyst, that stinging cell, finishes its maturation in the body of the nudibranch, which is, for any of you that know much about transplantation biology, normally our immune systems do not permit that kind of thing. And so in the book I talk about this as a model for transplantation biology because of the special ways that the nudibranchs do this. Now, maybe that Aeolidia, the shagrug nudibranch, didn't look scary. And I don't think that looks scary. This just came probably 3 weeks ago. This guy, it's called the blue dragon. It's gorgeous. This thing is shutting down the beaches, all right? And it is not shutting down the beaches with harpoons that it makes. It is shutting down the beaches with the harpoons that it steals from Portuguese man-of-war and other jellyfish. And then the thing I didn't even know, the venom actually becomes concentrated in the blue dragons, so they can be even more venomous than actually the Portuguese man-of-war. And so I just thought that was just an absolute perfect case in point. So I want to just take a couple minutes to talk about how this actually works. But actually, I've been fascinated by the blue dragon for quite a while. I don't have time to talk about it tonight, but my first book, A Sea of Glass, is about my work as a curator at Cornell with the Blaschka glass collection. And that version of the blue dragon there is glass, as are those jellyfish off on the right. Those are all made by the Blaschkas 180 years ago in glass. The thing that's cool is that's called the Blue Fleet. That's the Portuguese man-of-war, the By the Wind sailor, which we sometimes see on our beaches, and the blue button. And so that's where the blue dragon gets its venom and its nematocysts from, actually preying, finding. These things are in the open ocean, right? They're not even on rocks running around in the intertidal. They're in the way open ocean. The blue dragon somehow finds its prey, uptakes the nematocysts, concentrates them, and becomes a dangerous animal in its own right. And so a little bit about, there's a special word. We've got all these words for everything. Klepto means stolen. Nighty is the word for those stinging cells. So how does kleptonidy work? So here's our one of our nudibranchs, and this is a detail in one of those back projections, which of course has a name. I'm not going to tell you what that is because you don't need to know that. And so what happens is this is continuous with the digestive system, and so they get loaded into these cells and then mature up towards the end, at which point they're capable of being fired. And then, of course, they're dangerous. So here's a question. Here's a small trivia question for the group. So which of our local ones do this? Not all nudibranchs do it. It's not something that everyone can do. Does anybody want to hazard a guess about which ones in this picture would be the ones that do it? So you have to somehow tell me. Bottom right, top right. Bottom right? No. Top right, yes. So that's our Hermesenda, which feeds on cnidarians and does it. This guy fooled me for a long time. The beautiful Dirona with the white with the white stripes there. That's actually a whole nother family, and it does not do it. So it's only this one family of the nudibranchs, and there's hundreds and hundreds. It's probably the most diverse of all the nudibranchs, the one that this Hermesenda is in. And this guy eats sponges, and it uptakes all kinds of crazy chemicals from the sponge, but not nematocysts. And that's actually another little Hermescenda, so tiny you could barely see it. Some of them are very small. Just to sort of underscore a little more of my obsession with this, this is on my website. If you're interested in more materials, this is a blog I did for Friday Harbor Labs. We have a Tide Bites. It's a monthly kind of articles about natural history, and so I did one called a Nudibranch Life List. I thought it was perfectly normal to talk about having a Nudibranch life list. Until I mentioned it to one of my friends who's a birder, and she just looked at me and she said, "You have a what? What does that look like?" And I'm like, "Oh, it's just all the nudibranchs, that I've ever seen anywhere on the Pacific coast, north, south, in Indonesia when we're diving." She's like, "Well, how many are on that list?" I'm like, "Oh, I don't know." So anyway, she kept bugging me, so that's why I wrote this article, to kind of actually put some numbers on it. So if you like nudibranchs, you might enjoy that. So let's talk about sea stars. The superpower in the book that I talk about is the sea star's sticky skin. Oh, before I talk about that, I would like to just highlight one of the great joys for me in doing this book. I do have a crossover interest with invertebrate and ocean art, and so I was able to collaborate with an artist who I've long admired, Andrea Dingeldine, and she did the chapter opening diagrams for each of the chapters in the book. And I particularly love the sea star one because here's our friend, the ochre star here, sort of lording it over the intertidal and all the mussels. And then here's our sunflower star deeper in the water. Whoops, go back. And so I really do love her diagrams. So sea star sticky skin, what is that? It's got another big name. It's called mutable, which means changeable, collagenous, which means with collagen, tissue of echinoderms. It's a very big deal in the world of smart materials and the world of transplantation biology, because this skin in sea stars transforms from kind of loose and flaccid to very, very stiff at the firing of a neuron, so it happens almost instantaneously. And what happens is when that neuron fires, these are the collagen fibers in the skin, all of these cross-links line up and make it really very stiff. And so it's called a smart material because it's under electrical control. And the reason that's really an obsession with materials scientists is if something's under electrical control, you can add a different trigger, like light or temperature. And so there's this fascination with being able to change the stickiness of this skin with different triggers. So that's the what's in it for us. Of course, sea stars use it. None of you, I'm really pretty sure none of you can open a mussel. I actually had mussels for dinner tonight, but they were already cooked, so they were open. I would not have been able to open them. The way that stars do it is, of course, it's holding power. They attach their suction like tube feet. They hang on. They set the stiffness of their skin, which means it doesn't cost them a lot to hold that power for so long. And also the other bit about the sticky skin, why it's such a cool superpower, is probably most of you know that sea stars can autotomize their arms. They can drop an arm, and in some cases they can regenerate an entire star from that dropped arm. So again, it's that ability to quickly dissolve some of these connections and put them back together that allows them to do that. Okay, so let's talk about this. I was at Friday Harbor Labs in actually 2011 when we saw some beginnings of this outbreak, and then in 2013. And some of my research is disease ecology. So because I was a long-term ecologist in the Pacific Northwest, the notion that there was a disease that was wiping out our sea stars was immediately a big priority for me. So by the time this got going, it was the largest marine wildlife epidemic. started in 2013 to 2015. It continues on the West Coast, so we're still losing a lot of stars to this. Over 20 species were cataloged as being affected. The sunflower star, the one I'm shown here, experienced a 98% decline in the continental U.S. That was billions and billions, because some of you divers will remember they used to be as common as a robin in our waters, and you saw them on pilings, and you don't see them now very much, almost never. So through a series of papers, we sounded the alarm, and a particular concern about this one because it was the most susceptible of all the 20 species. So kind of like COVID, which affected not just humans, but also cats and minks and other organisms, this was a multi-host pathogen as well. And so I'm just going to tell you a little bit of a tale of two of our sailor sea stars, probably familiar to all of you, the ochre star, which was noticed right away at the start because it's all over our mid-intertidal. The sunflower star was one of the ones we got some of the first reports on. These were pictures from October 9th, 2013. And then about 20 days later, you can see all of these stars in that same rock were dead. So these were the kind of alerts that we were getting about these two dominant species, which is where we saw it initially. This is some of the data. My PhD student at the time, Morgan Eisenlord, led this study. And what we did is we went out to all of our intertidal sites in the San Juan Islands, and they're all different colors here. And what you can see is that by June of 2014, at some sites, 100% of the stars had symptoms or signs of that sea star wasting disease, which were the ones I showed you on the previous. There were lesions and their arms falling off. And then, of course, the numbers dropped so much that to date, we see it's actually quite rare. This is what happened to those populations from 2014 on. Some of the sites had almost 500 stars. Some of them had 250, 150, many, really huge numbers, and they just plummeted into 2015. And they've remained pretty low. One of the sites shown here, I was at this, there were 150 stars at the beginning, and I went out there this spring, and I found one. And that's our ochre star. So we are seeing some positive signs in the ochre stars. They're really doing pretty well at quite a few sites, but they're not back to where they were, and they are still showing some mortality. This is the data from the sunflower star, and we noticed it in 2014 beginning to go. This was the data from 2015 in Friday Harbor, and Mo Turner was our lead diver on this. I think this is really interesting data because look at May. This is the percent of those stars, these are the biggest in the world, right? They're big. The percent of them that showed any signs of the disease. And you can see in May, very few of them. It's like under 10%. By June, it had doubled. By July, it had doubled again. And then by August, virtually every one that we saw had high levels of the disease. And of course, a year or two later, they were virtually gone. And so that's what virtually gone kind of looks like. This is citizen science data from colleagues and citizen science divers. And it's kind of great data because it goes back to 2006. There were lots of sunflower stars, and there's no mistaking them, right? 24 arms and this big around, even the little ones. And again, they just plummeted immediately. Oops, sorry. Get this. There we go. Just plummeted, the numbers plummeted, and they have not returned. The thing that happened, these are sea urchins. The sea urchins began a big boom, and I'm going to talk more about that in a minute. And then the other interesting bit that I'm going to come back to is this other species of sea star. This is the leather star. That one increased, so it remained pretty much unaffected. And I'm going to come back to why that's really interesting now to us. So these are just, a few of the papers that we've published over the years. We published this paper using the citizen science data. And again, what you can see is this is just a sunflower star. Virtually every basin in our waters and all the way up into Canada, the same, and actually down to 500 feet in the deep ocean. So there was no refugium from this. So why do we care so much? Well, of course I care because I just care. And all aspects of the biodiversity are important to me. But this one is ecologically important because it eats a lot of urchins. And it is the coolest thing to watch them eat urchins. So we have a whole captive breeding program at Friday Harbor Labs for the sunflower star, and so we're always feeding them. And they swallow them whole and spit out the spines. And so this is a really impressive predator. And when it comes to the little baby urchins, they just vacuum them up. So when you removed all the sunflower stars from a habitat, the urchins exploded. And even just last week in the San Juans, the Department of Fish and Wildlife collected thousands of urchins to just get them out of the water because they were devastating our kelp meadows. This has been an even bigger problem in California. It's a picture from the Nature Conservancy, who's funded some of our work on this, showing what we call an urchin barrens, where there's no sunflower stars. So there's a lot of push, a lot of interest in recovering this species. Our team worked to get it listed as an endangered species with an International Union for the Conservation of Nature. I'm incredibly proud of the work Jason Hodin has done at Friday Harbor Labs in developing a captive breeding program, which has now been exported to all of the major aquaria on the West Coast, the Seattle Aquarium, the Monterey Bay Aquarium, the California Academy of Science. So workshops were run at Friday Harbor Labs to teach the captive breeding methods. Jason has started releasing sunflower stars in the San Juans, and that the whole point of this is to be able to eventually get them back in the water. But of course you're going, well, wait a minute. How can you release them if there's this thing that's killing them? And so this is, I want to tell you a little bit about our work on the epidemiology of disease. And this is our team. This is an international team of scientists, of me, and then a scientist from Canada with the Hackeye Institute, and then a PhD student at the University of Washington, Grace Crandall. And we worked at out near Port Townsend. And I'm just going to tell you a little bit about this paper that we just published about a month ago. Did anybody read about some of that? Yeah, there was actually a lot of pretty amazing press about the discovery of the Vibrio pectinacida as a causative agent of sea star wasting. And of course, we worked predominantly with the sunflower star because it's the most susceptible and the one that would be the most sensitive to being able to show us levels. And the way this work was done is challenge experiments in the lab. So stars were challenged with various kinds of active fractions from 6 stars, and then this was always controlled. This is the control, is the heat-killed version of it. And the amazing thing about these experiments with hundreds and hundreds of stars is that virtually all the controls survived, and all the treatments didn't. And this is kind of what it looks like, and I just want to give you a little bit of detail. This is a healthy sunflower star. When they get sick, when they develop this, the first thing we see is what we call arm twisting. And then to the point where they'll eventually just contort all those arms together into a ball, and then this is arm drop when they fall off. So in these assays, our team leader, Dr. Alyssa Gaiman, would keep track of when do they start to twist, when do the arms fall off, and then when do they die. And so in the initial experiments, just using water, so this is a waterborne pathogen. It took 10 days of exposure before the arms would begin to twist and fall off and die. They all died. And then over time, over the 4 years of this study, we developed more and more detailed methods. So this was using coalomic fluid, which is basically the blood of the star. And the thing to note here is that it only took 5 days for the arm twisting. So half the time when you used a more concentrated source of the infectious agent. And this is the way that Alyssa and team were eventually able to figure out that it was the bacterium that was in the coalomic fluid that was the killer of these stars. And then the cool thing here, I'm just so proud of this part of the study, is our team member was able to get this Vibrio into pure culture and test it to really show that it was that bacterium that was causing the mortality, and at different concentrations. So this is low concentration, and it gets worse and worse as you increase the concentration. And so that's kind of the kind of data that was in that study. And I'll be happy to answer any questions about that later. I've really skipped over quite a lot. It has been detected in stars, the sunflower star from Alaska during the earlier days of the epidemic as well. So what do we do? Okay, so this is great. I mean, it's really difficult to manage a disease if you don't know what the causative agent is. So now we're really excited about being able to move forward with the recovery plan. The first step is just to make sure to maximize genetic diversity. And a cool discovery is the ability to cryopreserve the larvae. That little thing in that droplet is actually a sunflower star larva. So again, we're breeding billions of them at Friday Harbor Labs. But they can also be cryopreserved, and that means it's a method that's widely used by conservation biologists to preserve genetic diversity. I mean, what if there's a new strain of this that comes out that's even worse, right? And we get more mortality. So that's the first step. The second step is to investigate the possibilities for selective breeding for resistance, and then being able to put more resistant stars out. As I said, some of these captives, the selective breeding is going to be a 5 to 8 year project. That'll take a while. But now that we know the agent, we can monitor it in the water and in the food these stars are eating. And then the other part of the work that we're doing is studying the immune response, because that will play into the selective breeding. And I'm just going to briefly show you some really cool results from Grace Crandall's work, her PhD work. So she tested these 3 species. That was the one in the figure I showed you a long way back, that was totally resistant. And so head-to-head, all 3 of these were exposed to the agent. And this is what happens. This is the sunflower star. Again, by day 20, they died. This is the ochre star. It's a little bit more resistant. It lived longer, and one of them even lived to the whole end, over 50 days. This is the leather star, totally bomb-proof. It's housed in the same containers and doesn't. And so what Grace is doing is studying the immune system, and what is it this one's got that our sunflower star does not have. And I'm going to wrap up here. This is just some of the work that we're continuing at Friday Harbor Labs, a focus on sort of the recovery effort. And I'm just going to end. Thank you for laughing. It's good nature. I love our terrestrial planet and all of our organisms on land, but in terms of evolutionary drama, I think of terrestrial life as a bit of a sideshow, because all of these major body plans and lifestyles and their supporting superpowers evolved so long before terrestrial life had progressed beyond non-vascular plants like liverworts. And so I hope you've come to see some of that. And then these are just some of Andrea's beautiful diagrams for the different chapter openings in the book. And I'm going to end with that and really look forward to taking any questions. all for listening and for laughing. And I think the instructions are that if you have a question, they really would like you to come to the mic because we're recording this. There are people online that are watching this, and they won't be able to hear unless you speak into the mic.
Thank you for your lecture. Given that billions of sunflower stars were exposed to the pathogen, it's a huge sample. Did any have any natural immunity or resistance?
Thank you. That is one of the million-dollar questions. We ran through, 270 stars in our experiments. None of them ever survived, but those were pretty high dosage. So now the effort is to go back and sort of look a little bit more carefully about whether there are survivors at lower dosage. There are still sunflower stars occasionally popping up in places, but they are pretty rare. I mean, they really got hammered. And it is possible for a pathogen to completely wipe out a species when it's a multi-host situation like this. So we don't quite know what the outlook is. There are some really exciting refugia in colder waters. I didn't even have time to talk about the climate links in this, but warming waters are a very bad thing. There are colder fjords in British Columbia where there are refuge populations.
Cool, thanks.
Yeah. Oh, good. Please come up.
Thank you for your wonderful talk. And as coming from a family of sea star enthusiasts, it really means a lot to me. So thank you. My question was about, I guess, the control group in the experiments that you presented. You mentioned that the water could be something that transmits the pathogen. And I wondered how you assured yourself that the water in the control group was free of the pathogen.
So we did all these experiments at Marrowstone Point, which is a USGS fish virology lab, and it was a really wonderful controlled location where everything was UV filtered and filtered before the animals were put in their water. So we don't know that the animals that were brought in had not been previously exposed. So that was actually why we were so happy that the controls survived as well as they did. And then we were really careful that they were never exposed to the sick ones. And anytime they were, everything had to be just euthanized because once they were exposed, that was it. Yeah.
Thank you again.
Yeah. And then the other part of it is that we were able to use some of the captive bred stars, which hadn't been exposed. And they basically behaved the same way. Everything was identical with those captive bred stars versus the ones in the wild. Yeah. Good question. Thank you.
Thank you.
Yeah.
Hi.
Hi.
Hi, my name's Annie. I also have gills behind my ears and love diving in our local waters. My question is, I understand the science that just came out that you published this last a few weeks ago. And where does this come from? Like, so was it not there before 2013, 2015? Like, what is happening? And the reason, like, can we test our water for
Yes.
this pathogen?
Yes.
Because, like, I noticed a dive site up north, Mukilteo, we still have active melting happening all the time in the water. So I'm like, well, there's outflow there. Is this coming from our outflow? Is this coming from us on land?
Yeah.
Where is that? Now that we know what it is, how
So
do we figure it out?
I mean, we've been flying blind, right, for the last over a decade with no ability to test or look or know where it came from or how it moves around in the water or where it gets concentrated. Now we can answer those questions. We don't have the answer to those questions yet, right? So this is the next phase of this work. Is certainly to look in places, and actually it'll be great to know about spots that are kind of local hot spots, because immediately we would like to test the water. We'd like to test the food the stars eat there, right?
Yeah.
So the clams and mussels could be concentrating this bacterium and delivering a lethal dose in some places. And the million-dollar question, and I know we're going to get an answer to it, but it may be a couple of years, is where it came from and whether it's actually new on our shores. It has been reported from Europe in scallop larvae, so it is known previously as a pathogen. So I can't even speculate about whether it's been in our water for a long time and suddenly a new strain emerged, or whether there was a new introduction. But it was a very dramatic onset of the epidemic, as
Yeah.
you may remember.
Yeah, completely.
That spread very rapidly, yeah.
Without somebody like you doing the work. So thank you so
Yeah,
much.
you're welcome. Somebody obsessed enough to keep at it for 10 years.
I'm Maxwell, and I'm just curious, why does the sea star wasting disease make the sea star's arms curl up and fall off? What part of it makes them curl up?
Yeah, that's a really good question, Maxwell. I wish I could answer all these questions. It suggests that there's some neural involvement. I mean, we do know that the bacterium is very abundant or present in the blood of the star and the coelomic fluid, but the twisting suggests there's some kind of neuronal environment as well. And so some more histological work to actually look carefully at those twisted arms and just see whether you have infected nerves causing that. But we don't know. Yeah, but it's striking because the sunflower star, they just kind of all crumple up together, but some of them actually all twist the same way. It's really weird. Like the ochre star, the 5 arms will all twist one way, and it's quite striking. Yeah. Good question, thanks. Yeah.
Hi, thanks
Hi.
for the talk. It was super awesome. I did want to say I did see a sunflower star about 8 big. It's the size of our data sheet in Hood Canal earlier this year, so they are there. I am curious about, in your research, if you've seen any obvious indication on what humans have done in climate change in terms of the wasting disease. I know you mentioned you don't exactly know where it came from, and the warm waters are obviously not helping. But if there was anything that we've done that has directly impacted or caused this.
Well, I like to say that the warming oceans really fueled the epidemic. So especially, the blob, 2015, and into that, late 2014, a lot of warming. That was when we first saw the sunflower star. We had a tank outside that overheated, and it immediately just showed signs of disease. So California it's almost completely devoid of sunflower stars because of the temperature, because the water's warmer. And of course, Alaska and British Columbia are more of a refuge. This is an endemic species. The only place in the world it lives is on our coast, used to be from San Diego to Alaska. Now it's kind of from Washington to Alaska, and the Californians are really determined to try to get them back in their waters, and we hope that can happen. But temperatures it's like we're constantly fighting warming, and especially with disease. Yeah.
Thank you.
Yeah. Thanks for your sighting. That's exciting. Yeah.
When the sea stars, when the sea star's arms fall off, why does it kill them? They can regrow arms.
Well, that's a very good question. So it doesn't always kill them. So sometimes we'll find stars that are growing back arms, and so maybe they're in a hot place, or it's August or September. They're losing their arms, and then the water gets cold, and they pull through it, and then they can grow back their arms. So it's not the loss of the arms that kills them. That's just a sign that everything's kind of going badly for them.
Okay.
Yeah, good question.
Again, I will say thank you for the talk, and it's very fascinating. I'm just curious, how did you get into? I always like to ask people how they found this lifelong passion, and you still do it and stuff. So I'm just curious what led you to be, what you wanted to do with the rest of your life.
Yeah. Well, I don't know. I guess it goes back to childhood. I mean, I love nature. I used to be a frog hunter as a child. I used to go catch frogs, and I was fascinated with the diversity of different species of frogs. I don't know. And then it turned to be the ocean. So even, as a college student, I was completely fascinated with invertebrates in the ocean and life in the sea. And then to be able to have the opportunity to be a saturation diver on a research project as a grad student, that definitely sealed the deal for me. Just that opportunity and to just see the magic of those waters all day and all night, and just the magic. So I don't know if that answers your question. And I've also loved educating. It's been an incredible thing to be a college professor. I've loved doing that. Yeah.
Thank you.
Yeah.
We have a few questions that have been submitted digitally. Are there any citizen science needs or opportunities around sea star wasting recovery currently? Anywhere we should be submitting Salish Sea sightings?
That's a really good question. We're not currently working actively. There is a group. There is a group in California that's still monitoring, and they may be taking some citizen science data. They were for a long time. As far as I know, they're not currently. Reef would be. Okay, thank you. Thank you very much, Dr. Parrish and others. Yeah, reef and reef check. So are they taking data on lesioned animals?
Yeah, so I just did the reef check training this year, and we are these are all the things that we're counting. I'm not 100% sure where all the data is going right now, but if you're a diver and you can definitely be submitting data through reef.
Great.
Reef check is a little more particular, but reef is a pretty solid one as well.
Great, thank you very much. And Dr. Parrish here also runs a seabird citizen science program. And early in the epidemic, their team actually helped us and sent a lot of the early observations to us. So we're really grateful. And I'm not sure that we would have ever identified the extreme hazard that the sunflower star was in if it hadn't been for the reef and the reef check data, the citizen science program. Yeah, good question, thanks.
What are the 3 coolest things about sea stars that you haven't mentioned yet?
The 3 coolest things about sea stars, their colors, right? Oh my God, every time I see just a pile of ochre stars and all those colors, and I just wonder, what are they doing with those colors? So colors. What else is cool about sea stars? They're like Martians, right? They're not normal, obviously not normal animals, but yet they're one of the most voracious predators, top predators in the ocean. And so I think it's just fabulous, the sophistication of them as a predatory killing machine, if you're a clam or a mussel, or some other things. Some of them eat anemones, so that's 2 things. Oh my God, what's the 3rd thing? How fast they move, right? I mean, the sunflower star, it moves that fast. And so, yeah, nice question.
Are there other strains of that bacteria or that family of bacteria that are affecting other echinoderms? Like, are there any that are affecting sea urchins, or is it just the sea stars that are affected?
It was just the sea stars. I mean, early in the epidemic, we really looked hard. We were really worried about the sea cucumbers and urchins. And it was very clear that it's the group of the stars that we call the asteroids, the asteroidea, and not brittle stars either. So yeah, very specific for asteroids. Yeah.
And follow-up question to that, is there anything about the structure of sea stars that make it more susceptible to this disease, or is it just that this particular bacteria doesn't really affect any of the other echinoderms?
Yeah, I don't know. That's a really good question. We just don't know what it is about why they're susceptible, but scallops are also susceptible to this same species, and it's a nasty, I mean, probably people know that there are a lot of Vibrios out there, right? I mean, the clams and oysters, you have to be careful about Vibrio parahemolyticus, Vibrio vulnificus. Those are deadly to humans, make humans sick. So this is a completely different species. It's not like related or similar to those in any way. But it does create, it makes a very nasty toxin. And it's a weird thing. We did a lot of histology early on, and we never could see it. And the scallop people reported the same thing. Somehow that bacterium just kind of gets in there and sort of disappears and goes invisible. Yeah, so there's a lot of, I mean, I'm getting excited now. There's a lot of really interesting biology to understand in that host-pathogen interaction.
Thank you, and thank you for your talk. It was really cool.
Thanks. Yeah. I think we should probably end the, oh, come.
I remember, I grew up in the subtropics, and I remember years ago, crown of thorns starfish being rampant and out of control. And then, you know, it sort of disappeared from the topics, and I'm wondering what the status of the crown of thorns, and is it just because they ate all the coral, or what's happened to them?
Crown of thorns are still a really big problem, and there's places in Australia where they're just pulling them out of the water. They're very destructive. Yep, and as far as we know, they're not susceptible to this disease. Yeah, figures, right? So I do think we should probably stop, because I think I'm going to sign any books that people have, and so we have other things to do. But thank you so much for your interest. Yeah.